All right, thank you. Moving on to our next speaker, it's my pleasure to introduce Dr. Robert D'Amato from Boston Children's Hospital. Dr. D'Amato's work is largely geared towards drug development and anti-angiogenic therapies, and today he's going to be talking about using genetic context differences to identify modifiers and mechanisms of angiogenesis.
Okay, well, thank you for the invitation. I want to flip from talking about diseases of blood vessels of small blood vessels to talking about diseases by small blood vessels, i.e. angiogenesis in pathological situations. I want to start by showing a slide from a paper by Dr. Folkman in 1996 where he introduced the concept of angiogenic balance. And that is that locally, small blood vessels are responding to the angiogenesis stimulators and the angiogenesis inhibitors that it sees. So there's angiogenesis stimulators, VEGF, FGF. There's angiogenesis inhibitors, thrombospondin, platelet factor 4, angiostatin. And normally the balance is tipped way towards inhibition. The vascular system is quiescent. But under certain pathological situations, such as a tumor, you have upregulation of VEGF, and the stimulators overwhelm the inhibitors, and the inhibitors are also downregulated in many of these circumstances. So we've been interested in studying the angiogenic balance, and we use a model called the corneal micropocket model, where we place a pellet of growth factor in the cornea of a mouse, and we measure the blood vessels that grow. And I developed this model about 20 years ago. We used it to screen angiogenesis inhibitors, and we also were interested in looking at strain differences. And here you'll see two different strains of mice, the C57 mouse and the 129 mouse. And the same pellet is being implanted in both these two strains of mice, and you can see such a drastic difference between them. And we were frankly fairly shocked by this because we assumed that angiogenesis was so tightly regulated in terms of development of the placenta and the embryo that there wouldn't be this level of difference. In fact, you have to put a 10-fold higher pellet of growth factor in this eye to get the same amount of angiogenesis as you see in this eye. So it's a really very large difference. I might note just about this model, the C57 black mice that we use routinely for this model. We found that the effect on angiogenesis is not age dependent, so young and old mice have the same degree of angiogenesis, and it's not sex dependent, so we don't see big variation between male and female in this particular strain of mice. So there's two ways to tackle this. One is doing linkage-based studies, others doing what's called GWAS-based studies. We do both of these. I'm going to show you some data from a linkage-based study where basically we take a low angiogenic mouse, the C57, and we cross it with a high angiogenic mouse, the 129, generate F1s, take those and cross them one more time, and you end up with a scrambling of the genes between them so that you can track the genes that are inherited and which ones are associated with a high or low angiogenic response. The disease, the angiogenic responsiveness is a complex trait, much like the diseases that are angiogenic in humans, like macular degeneration is a complex trait, meaning that there's 6 to a dozen genes that we see that are responsible for the trait, each one contributing 10%, 20% to the overall phenotype. I'm showing you data on this particular slide from linkage we got to chromosome 7 with this particular cross. And chromosome 7 in a mouse is interesting because it harbors the pigment genes, tyrosinase and others. And this got us a little interested in pigmentation. And indeed, when we did fine mapping, we found 2 genes involved in pigmentation, the pink eye dilute gene and tyrosinase. Both of them had small effects on corneal angiogenesis, and we published that over the last couple of years. And that brings us into a discussion about pigmentation. So the disease that I work on, macular degeneration, is a disease where blood vessels are growing from the choroid circulation, which is underneath the retina, and these small blood vessels grow up into, break through the membrane and grow underneath the retina and bleed and cause vision loss. The other vascular bed, the retinal vascular bed, is damaged in diabetes, and in diabetes you get bleeding on top of the retina. and these vessels are not growing in the pigmented part of the eye. So in terms of pigmentation, we're really focused in on macular degeneration. And one of the things I'd always observed clinically, and is well known, is that African Americans have a much lower rate of choroidal neovascularization than Caucasians. As well, working at Children's Hospital, you see that infantile hemangiomas is really a disease of young, white, blue-eyed babies. It's very rare in African Americans. So there is some pigment regulation of angiogenic diseases. So we decided to look at this by looking at keeping the genetics constant, by looking at C57 mice, but looking at albinos, albino C57 versus black C57, where there's a spontaneous mutation in tyrosinase. And what we found is that there was small differences in the corneal neovascularization, but gigantic differences in iris neovascularization. So the pellet was releasing growth factor that affects the front of the eye and leaks onto the iris. And here you see in the pigmented C57 animal, there's new vessels growing on the surface of the iris. But in the albino animal, there's massive angiogenesis, and that results in this big blob of blood that's in the eye, which is from these new vessels that are very fragile and they bleed. So here we're able to mimic that phenomena, and we were pretty convinced that what we're seeing is that pigmentation was somehow inhibiting angiogenesis, and we just had to find whether it was melanin that was inhibiting angiogenesis or something else that was inhibiting angiogenesis, and that's why African Americans are protected, because they have this inhibitor. But to our surprise, what we found is, yes, there is a difference if we take conditioned media from Caucasian melanocytes versus African American melanocytes. There is a difference. The African American melanocytes have less inhibition, but all of them are much higher than baseline. And to our surprise, what we were able to elucidate is not that there's an inhibitor being made by African Americans, is that there's a stimulator that's made by Caucasians that's in very high amounts that is in lower amounts in Caucasians, and I'll show you that. So by doing microarrays of messenger RNAs, we were able to identify a protein that was expressed in very high amounts in albino melanocytes that wasn't present in pigmented melanocytes, and it was a secreted protein. This protein is called fibromodulin, and fibromodulin is a member of the leucine repeat protein family, which also contains famous proteins like keratin sulfate. And it's a small secreted proteoglycan which interacts with other collagens and has some signaling through TGF-beta. So we went back, and after we'd seen this on the microarray, and we looked at non-pigmented melanocytes, albino melanocytes, we see very high amounts of fibromodulin in these melanocytes, these mouse melanocytes, versus pigmented melanocytes, we see very low amounts of fibromodulin. And we've looked at human melanocytes from culture from skin of Caucasian versus African Americans. We see very high amounts of fibromodulin in the Caucasians versus African Americans. So we wanted to see whether fibromodulin really is angiogenic, and we've tested it in every model we have in our lab. We've looked at in vitro models like migration of endothelial cells. We've looked at corneal angiogenesis, just putting a pellet of fibromodulin in the eye at the same dose as a VEGF pellet is able to induce more angiogenesis than VEGF. We've looked at it in choroidal neovascularization. So if we do a laser burn in the retina of a mouse and we add fibromodulin, we see a marked potentiation of the angiogenic response. So it does have in vivo angiogenic activity. And lastly, in this paper that we published in JCI this year, they wanted us to show that we could knock it out, so we used fibromodulin knockouts that were albinos. So this is a normal amount of, we're looking at the surface of the iris after a pellet of growth factor is placed, and you see that tremendous angiogenesis that I showed you before, and if you knock out fibromodulin, you see a marked drop in angiogenesis is quantified here. So this is our model for the pigmentary effects, which is that in a albino or low Caucasian with low amounts of pigment, there is very high amounts of fibromodulin being made, which shifts the angiogenic balance. Not that you're getting angiogenesis spontaneously, but you're tipping that scale, making it easier to induce angiogenesis. Of course, there may be evolutionary pressures for that. If you have a high angiogenic balance, you're going to heal wounds easier. So if you're bitten by a tiger, you're going to heal that wound easier, but you're also going to be more prone to getting a tumor. So there's a plus and minus to shifting your angiogenic balance one way or the other. And on the other side of it, pigment individuals have very low amount of fibromodulin being produced and are lower angiogenic, and therefore, if in that setting, you're going to be less likely to get cancers in pigmented tissues. I wanted to just touch on one other subject because we're talking about broad topics here that are needed in the field. But it's important for us clinically to be able to tell who is going to get pathological angiogenesis. If we could know, seeing a patient, that they're at very high risk of getting choroidal neovascularization, we could treat them differently and screen them differently. But currently there is no method to do that. We have seen some correlations. So when we look at the low angiogenic mice that I showed you before and the high angiogenic mice, if we look in their blood, we see that there's a correlation between circulating endothelial cells and circulating endothelial progenitor cells, such that the high angiogenic mice have high levels of progenitor cells and circulating endothelial cells in their blood. As a correlation, we're not saying it's causation. I don't think that these high endothelial progenitor cells are causing them to be high angiogenic, but they're just associated with that. And so one possibility is that we could measure these cells in the blood of patients and tell whether you're a high angiogenic person, susceptible to getting cancer reoccurrence, susceptible to getting choroidal neovascularization, if you have the risk factors. On the other hand, the problem with these kind of circulating cells is there's many studies showing that, for instance, statin use can influence the levels of these. If you have cardiovascular disease, there's depletion of these cells. So anytime you're looking at any kind of circulating marker, you have to take into account what drugs the person is taking, what diseases the person has that might be altering some of these factors. But this is one of the biggest deficits in the field, is not having reliable markers to measure angiogenesis and angiogenic potential. It's important in cancer trials as well, because we're trying to measure what kind of response we're getting to angiogenesis inhibitors. Are the angiogenesis inhibitors working? Do we have to wait until the tumor goes away to know, or can we measure the response in a very early part of the therapy and tell whether or not the person's responding by a drop, and for instance, drop in their progenitor cells? So that leads to sort of my lessons learned and some gaps. So the lessons learned is that there are significant variations in angiogenic potential that's driven by an individual's genetic background. And these variations are controlled by polymorphisms in genes. In the case of one of these groups of genes, pigmentation, that tyrosinase and pink eye dilute in mice regulates angiogenesis. And we've identified and are going to be publishing other classes of genes that contribute to the complex angiogenic trait. In the case of pigmentation, low levels of pigmentation are associated with high levels of angiogenic disease in pigmented tissues, so in the skin, in the iris, in the choroid. Proteins such as fibromodulin, which are secreted by melanocytes, are potentially important as controlling this pigment-related response, and responsible for why Caucasians have a higher angiogenic potential. And lastly, that circulating endothelial cells may serve as a marker for angiogenic potential, but really there's this need to be able to measure this so that we can tell which of the patients. Only 10% of patients that present to us with drusen and dry macular degeneration go on to the wet form of the disease and actually have severe visual loss. So we could figure out who's in that 10% group, would dramatically change our therapies in ophthalmology. And so I conclude with the gaps, which is there's numerous unknown polymorphisms that are yet to be worked out, and the downstream protein changes that regulate angiogenic potential. And we need to identify in animals and then validate those in humans to see whether or not these same genes and the same proteins that regulate angiogenesis in animals are also present in humans. If we have that, then we would be able to develop a panel of genetic tests. We could come in and take your blood and say, you've got these mutations. You have a mutation in the thrombospondin receptor, and that makes you a higher angiogenic person. Or you might have a mutation in the VEGF receptor that makes you a lower angiogenic person, and then understand how that's going to affect your susceptibility to angiogenic diseases. And of course, for all this to be have an impact, we need anti-angiogenic therapies which are safer, and which can be given before angiogenic diseases develop in a more preventative approach than we're doing now with more of a therapeutic approach with drugs like Avastin. Thank you very much.
All right, thank you for that beautiful talk. I'll take questions.
Hi, Jan Kirievsky from Columbia. Inflammation in macrophages, is the microphone on? Yeah. Inflammation in macrophages can influence angiogenesis.
Tremendously.
Have you thought about looking at the different macrophage response?
Yeah. So the question is inflammation in macrophages, and macrophage inflammation, and wherever there's angiogenesis, there's inflammation. You never see them separated. ICAMs are upregulated. You get macrophages plugging. Macrophages are loaded with VEGF. And so when we do a lot of these things, we do go in and validate them in skid mice and try to deplete macrophages and radiate animals, and we've done that. But many of these polymorphisms that are controlling angiogenesis are going to be in the inflammatory pathway. We haven't identified any of those yet, but they're hand in hand. Yeah.
Hello. Here.
Yeah.
Hello. So about the what you mentioned, the intriguing idea about the circulating endothelial cells.
Yeah.
There is a lot of controversy about what are the markers to identify those real endothelial progenitors or with potential angiogenic functions.
Yeah.
Could you comment a little bit on this, and how about the origin of these cells? Are progenitors or endothelial cells that are residing within the vessels, and upon an angiogenic stimulus, they just circulate in the blood?
So I'm of the camp that I've studied this. I do not see circulating endothelial progenitor cells contributing directly to vessel growth. I mean, in terms of incorporating into the endothelial lining. I see when I inject endothelial progenitor cells into mice, I see them going to active areas of angiogenesis, but into the stroma as supporting cells. I don't see them contributing. So I disagree with the notion that these are necessary for new vessel formation in terms of. The second part of your question is local progenitors versus circulating progenitors. I think probably local progenitors are more important than the circulating ones. But I'm just saying here that it's a marker. For some reason, circulating endothelial cells are higher in the angiogenic mice. I don't think they cause I don't think they are responsible for the enhanced angiogenesis in those animals. Yeah.
Yeah, it's very interesting. A simple question: have you looked at the brains and in any kind of angiogenesis model?
No, I've only I'm an ophthalmology lab, so we've looked at all over the eye.
Okay,
but we
so
haven't looked
brain
at
is out of
but it's
question.
easy enough to do. Just get yourself 129 mouse and compare it to a C57 mouse, and you'll have your answer.
Yeah, a lot of people use albino mice, so I'm just wondering,
But the pigment effects are predominantly in pigmented tissues, so in the eye and the skin and those kinds of things. So but yeah.
Thanks.
Hi, Chair.
Two small questions. The first is retinal pigment epithelium underneath the retina is pigmented in pigmented animals, and fibromodulin is, as you say, diffusible. Do you see RPE make this molecule?
Yeah, we do see a little bit, but we see much more in the uveal melanocytes than we see in the RPE. I don't know, but primarily because we think it's tyrosinase regulated, and there's much more tyrosinase in the melanocytes than there is in the RPE.
And as you mentioned, retina is not pigmented in any animal. But have you looked in the OIR model, oxygen-induced retinopathy, for retinal neovascularization in your knockouts?
No. The only thing we've done in the retina is we've added fibromodulin into the eye and looked at development of the retina and showed that it affects development.
Oh, it does,
But
yeah.
but that's exogenously added fibromodulin.
Thanks.
And I would point out on the retina side of it, that diabetic retinopathy is worse in African Americans instead of being better, like CNV is better in African Americans. So the retina is not protected by pigment. I mean, the surface retinal vessels are not protected by pigmentation, whereas the choroidal vessels are. So that sort of fits with what we see.
Yeah. Thanks.
We have time for one more question.
So just a follow up on that question. So in the diabetes patient, so they have a new diabetic retinopathy, and they have a peripheral limb vascular regeneration problem. So
Yeah.
they have this kind of paradox. So what you think about you finding in the eye is also occurred in the human patient, diabetes patient, that they will have opposite role, or have you ever tried that?
Well, I think because diabetes affects the surface retinal vessels, which are not adjacent to pigmentation, we have not seen any difference in the surface retinal vessels. And keep in mind, this is in pigmented tissues. So if you're talking about peripheral vascular disease, it would only be in the skin that you would see these differences, not in muscle or nerve, causing neuropathies or something like that. So it's really the pigmented tissues that are regulated in this particular by this particular set of genes and proteins. Yeah.
Okay.
All right, thank you very much. We'll have to continue the discussion in the panel later. Sorry.